Riset Rekayasa Dr. Ratih Asmananingrum. Yang kami hormati para narasumber hari ini, Dr. Eunike Mcdowan dari Lund University Swedia, dan Profesor Dr. Eni Sudarmonowati dari Pusat Riset Rekayasa Genetika BRIN. Yang kami hormati moderator yang akan memandu acara hari ini, Dr. Andri Wardiana, serta para peserta Friday Scientific Seminar, Sharing Seminar seri ke-16 yang berbahagia. Assalamualaikum warahmatullahi wabarakatuh. Selamat siang, salam sejahtera bagi kita semua. Salam Om Swastiastu, Namo Buddhaya, salam kebajikan. Alhamdulillah kita panjatkan puji syukur kepada Tuhan Yang Maha Esa karena kita dapat berkumpul untuk mengikuti acara Friday Scientific Sharing Seminar pada hari ini, Jumat 17 Maret 2023. Bapak Ibu, mengawali acara pertama yaitu sambutan oleh Kepala Pusat Riset Rekayasa Genetika BRIN, sekaligus membuka acara kepada Ibu Dr. Ratih Asmananingrum. Waktu dan layar kami silakan.
Terima kasih. Selamat siang, good afternoon everyone. I think in this seminar we will have a bilingual communication, so we can talk bahasa Indonesia or English because our speaker today, Dr. Nisa, also speak bahasa Indonesia. She said that sudah mulai jarang menggunakan bahasa Indonesia, but your bahasa is still perfect, I think. So on this happy day, we can finally meet again at Friday Scientific Sharing Seminar. Actually, our seminar is held regularly twice in a month to share biotechnology and also bioengineering research experiences. So we have various speakers from various institutions such as university, ministry, industry, and so on. From this event, we hope that we can initiate the collaboration in research and also in capacity building. So in Research Center for Genetic Engineering, currently we have 3 research clusters, red, white, and green biotechnology, that focus on biopharmaceutical, molecular diagnostic, genetically improvement of plant, and so on. So from this today's seminar, we will have 2 speakers actually from Lund University, Dr. Eunike, thank you for accepting our invitation. Dan juga dari Pusat Riset Rekayasa Genetika, Profesor Dr. Eni Sudarmonowati, yang akan nanti memaparkan juga mengenai pengalaman beliau ketika menerima funding dari Fulbright di University of Rutgers Saya sebenarnya sudah banyak mendapatkan laporan dari beliau mengenai aktivitas beliau di sana yang tentunya sangat padat sekali dan sangat produktif, yang tentunya nanti juga dapat disampaikan kepada kita. Jadi melalui seminar ini saya harap kita semua dapat belajar banyak, sekaligus juga dapat menginisiasi kolaborasi baru begitu mudah-mudahan nanti ada sesuatu yang bisa dikerjasamakan dengan Dr. Eunike dari Lund University, atau setidaknya kita bisa sharing di sini pengalaman kita masing-masing seperti apa supaya kedepannya dapat menguatkan kompetensi dari periset kami begitu. Saya juga mengucapkan terima kasih, thank you for the committee and also for the moderator today, Dr. Andri Wardiana, and also the committee of the Friday Scientific Sharing Seminar yang sudah bekerja keras untuk melaksanakan seminar ini. Jadi terima kasih banyak Bapak Ibu semua, enjoy the seminar, terima kasih, salam sehat selalu, Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Terima kasih Ibu Rati atas sambutannya. Baik, Bapak Ibu, selanjutnya kita akan foto bersama. Mohon Bapak dan Ibu untuk mengaktifkan kamera dan kepada panitia untuk dibantu melakukan tangkap layar. Saya akan menghitung dari slide pertama. Kita mulai. 1, 2, 3. Layar kedua. 1, 2, 3. Dan layar ketiga. 1, 2, 3. Baik, terima kasih. Petiklah mangga, taruh di peti. Jatuh dua menjadi terpencar. Saya ucapkan selamat menikmati acara Friday Scientific Sharing Seminar. Baik Bapak Ibu, acara selanjutnya yaitu pemaparan materi yang akan dipandu oleh moderator Dr. Andri Wardiana. Kami silakan.
Terima kasih Mbak Andalusi. Selamat siang semuanya. Assalamualaikum warahmatullahi wabarakatuh. Good day everyone and good morning in Milan. Ya, masih jam 7 pagi ya. Ya, jadi seperti tadi disampaikan oleh Ibu Rati dan juga oleh MC, kita ada dua pembicara untuk siang ini ya. Biasanya pagi dan hari ini siang. Yang pertama ada Dr. Eunike Mcdowan atau biasa dipanggil Nisa dari Lund University, dan Ibu Profesor Enis Darmanawati nanti di sesi kedua. Nanti mungkin setelah presentasi langsung tanya jawab dan nanti dilanjutkan ke presenter yang kedua. Selanjutnya mungkin sekarang mulai untuk presenter pertama. Sebelum memulai, saya akan membacakan sedikit CV dari Dr. Eunike Mcdowan atau Nisa. So she is a postdoctoral fellow in the Division of Hematology and Transfusion Medicine at Lund University. She received her PhD from the Australian Institute for Bioengineering and Nanotechnology at the University of Queensland, Australia, in collaboration with the Australian Red Cross Lifeblood. Nisa has worked in research involving blood group genotyping, patch display for prophylactic anti-D, and now in blood group gene regulation. I think she did bachelor and honors degree from Queensland University of Technology, right? So mungkin kita bisa lanjut ke pemaparan materi. So Nisa, time is yours.
Thank you. Thanks, Andre. Thank you, everyone, for having me today, and I really appreciate the invitation. So I will just share my screen. So as I said earlier, just put this on. As I said earlier, this is something that I'm really passionate about in the transfusion lab, and what I'm really think that as a person who's developing monoclonal antibodies in this field should be aware of. So I think we all part of the biologics industry. A big portion of it is made up of monoclonal antibodies, and it's a multi-billion industry, as you can see from this graph on the left with the graph, and it's forecasted to grow each year that comes with 2024, reaching $200,000 million in sale forecast. And I think, and so what this article said was that it's forecasted to grow, and also not only that, but this is the breakdown on the right graph of what those monoclonal antibody therapies comprise of. And a large portion of them, you can see, is actually from oncology at the bottom, and that's actually looks forecasted to grow in 2024 and will probably take up most of the market. So I'll be focusing on two monoclonal antibody therapies in this part of monoclonal antibody therapies, and also their and how that affects pre-transfusion testing. So the first one is called daratumumab, which is an anti-CD38 antibody. It's a human IgG1 transgenic, made from transgenic mice, and there's multiple mechanisms of action. It's proven to be quite successful with complete responses, and 65% of patients remaining progress-free for 12 months. It's from Genmab, Jazz Biotech, and it's had its USA FDA approval since 2015 for the treatment of multiple myeloma. But it also has an orphan drug status for cancers where CD38 is overexpressed, as for example, B-cell chronic lymphocytic leukemia, which over CD38 overexpression is associated with prognosis. So those with high CD38 expression are more likely to have, die, basically. So yeah, so it's a and so they've used this and they found quite good results with that. And so just this diagram on the right shows how daratumumab works. It's by binding onto the CD38 antigen on the myeloma cell. And then it recruits macrophages to phagocytize the tumor cells, or natural killer cells, or effector cells to lysa cells, and or even just cross-linking with antibodies to induce programmed cell death. So that's quite a targeted therapy. And it's actually one of the highest selling in the top 50 of the monoclonal antibodies that's selling. And so the second one is rather new, and it's called magrolimab, and it's an anti-CD47 antibody. It's in the phase 2 clinical trials at the moment, but it's used to in myeloid malignancies, and so CD47 provides the "do not eat me" signal on the to the immune cells. So if we and something that's overexpressed in myeloid malignancy, so we can see this how it works in the picture on the right shows how it works. So if CD47 is expressed, it tells the macrophage, don't eat me, basically, I'm fine. But like if you block that signal from CD47 to the signal regulatory protein alpha, you have the macrophage eating up the tumor cells. So yeah, where tumors that have CD47 overexpressed, this is quite a good way of eliminating the tumor. That's currently in phase 2 clinical trials. So it's a humanized IgG4 monoclonal antibody, and it's most commonly used in acute myeloid leukemia and myelodysplastic syndrome. Okay, so what these two monoclonal antibodies have in common is that they also react with red blood cells, and then that's what causes a bit of a problem for us in the blood banking laboratory when we do the pre-transfusion testing. So pre-transfusion testing is basically the testing process we do to determine the patient's blood type and find compatible blood for transfusion. But it's for patients undergoing these therapies. These monoclonal antibodies are also in the plasma alongside all the other antibodies that they have. And so that makes our job quite difficult. So what our job is in the lab, I've broken it down to these 3 parts, I suppose. It's very simplified, but it's a bit more, it's like this. So you have the patient comes in for, they need a blood transfusion, and people with hematological cancers, they often do. It's not uncommon. So you do get these patients who do have a blood cancer, or like they would have a low hemoglobin or whatever as well, because I guess it would affect their bone marrow, the development of their cells in the bone marrow. So they would be getting blood transfusion as well. So we get the blood sample and we find out what their blood type is. We look at ABO and RHD, for example, and if they need to, we can look at other blood groups as well. The second part we do is we do an antibody screen. And so we test the patient's plasma for any unexpected antibodies. So any antibodies that could react with any red blood cell. So especially important if they've previously received a transfusion, or if it's in case of a female, they've had a pregnancy. And then also the third part is a cross match. So once we know the patient is patient's blood type, and we know that they don't have any we know the antibodies that they have or don't have, we can test the plasma with the red cells that we're going to transfuse, and we see if there's a reaction. And so what the reaction, so what's common between all these three tests is that we're testing the patient's plasma against a red blood cell. But if we have DARA or daratumumab or magrolimab in the patient's plasma, that's gonna just mask everything and react with everything. So like the best basis of the tests that we do in pre-transfusion. So we just get the red cells, the antibodies, and then we look at the agglutination under a microscope, and we also see if the antibodies can have complement activity and have lysa red cells. That's in a nutshell what we do. And so this is an example of one of the tests that we do in pre-transfusion testing. This is, okay, so if we look at the first line, it's called an indirect Coombs test. It's where we react the patient's red blood cells and the patient's serum, and this one doesn't have any antibodies in it. So then the red cells are not coated with anything, they're free, but then we test it with an Coombs reagent, which is an anti-human IgG antibody, and there won't be any agglutination. So we have a negative test result. Okay, so in the next part, we have another indirect Coombs test scenario where we test the patient's red blood cell. I will just turn on my pointer. I guess I can do that. Makes it easier so you guys know what I'm pointing at. So we have the patient's red blood cell, and we have the patient's serum that contains antibodies. So we tested the patient's serum against other red blood cells in the lab to see what antibodies they are. And then we would detect any bound antibodies on there with the Coombs reagent, which is an anti-human IgG. And then that would agglutinate and form a red blood cell clump. And we would see that in the column card. And so we would visualize agglutination. So for patients on daratumumab, for example, they would have those antibodies containing the serum. And then, so we test it against our own, we test that plasma against red blood cells that we use to figure out what antibody it is. And it just binds to all those red blood cells, no matter what blood group phenotype it is. And that's what we call pan reactivity. So pan being short for panel. So the panel reactivity means it reacts with all the red blood cells in the panel. And so that makes our job difficult, because then if everything is positive, then it's all a false positive, and we don't actually know for sure what antibodies, if the patient has any blood group antibodies there. And so we don't know how to confidently call the result, and we don't know what to transfuse the patient because we can't do our testing. Okay, so I'll just focus on the effects of, so this part I'll focus on daratumumab, and also yes, these are challenges, but these are challenges that we've found ways to overcome. CD38 is actually, yes, it reacts, but it's weakly expressed on red blood cells. And this group found that they can resolve the interference in that with daratumumab and with CD38. So if we look down at this table here, there have 5 patients in the study which were under daratumumab treatment, and this is their age and how much daratumumab they received, and also the period from when they last received DARA to when the blood banking test was performed. And so the result of the antibody screen and the antibody red blood cell panel was pan-reactive, everything reacted. And the DAT is the direct antiglobulin test, where they just see if there are antibodies in the patient's plasma that reacts with their own red cells, so like any autoantibodies to the red cell, and that they do positive, which is actually expected because CD38 is also on the red blood cells. But I guess in some cases with this one where it's negative, there was a longer period between this, when the DAT was performed and when DARA was given. So, but like what they found was when they used DTT, so that's a common reagent that breaks disulfide bonds, it would affect the antigen on the red blood cell, and so CD38 doesn't actually bind to the red blood cell anymore. So, so that's a good way of navigating around it and troubleshooting DARA interference, so DARA for short. But the issue is that, and so the issue with DTT treatment. Okay, so I'll just go, sorry, this one. Figure B, that what they've done is that they've looked at this like loss of antibody reactivity for DTT treated red cells, and they found that, yep, this is a graph showing the DTT concentration and then the DARA that was added. So this one on the left doesn't have any DARA or DTT. And then the x-axis shows the percentage of DARA binding. So we get high DARA binding without DTT, and with increasing concentrations of DTT, found that there was less and less daratumumab binding. Okay, and then also another common reagent that's used in the blood bank laboratory is trypsin. So we use these enzymes commonly to destroy antigens, and so that's how we can figure out the specificity of certain antibodies. So if we enzyme treat and then the antigen's destroyed, and then we also lose the antibody's reactivity, that means we've found that antibody was specific for that antigen that was just lost. So this one is a one showing for trypsin, and it shows that also trypsin concentrations at 1 or 2% remove or reduce daratumumab binding. The issue with using DTT and trypsin is that it also destroys other antigens in the red blood cell. So I think most of them, this graph shows which ones are destroyed by DTT and trypsin. Papain-Physin is another one, but we won't go into that. And I think the biggest concern with DTT is that it destroys KAL, and KAL is, I think, probably one of the most clinically significant blood group antibodies. So that would be the main concern. What they found is probably another way you could remove this interference from DARA is to have an anti-DARA idiotype. So an anti-DARA idiotype, it will just bind to all the free daratumumab sites in the plasma, and then it would just block the antibody from binding to red cells during our testing. And so in this graph, you would see daratumumab binding almost 100% here. And then when you add the anti-idiotype, blocks all the anti-DARA sites, and then that reduces it greatly. compared to the isotype control. So I think that's another way, if you're really looking into the Cal blood group system or an anti-Cal, you can find your way around it with an anti-Dara idiotype. And that anti-Dara idiotype was actually provided by the manufacturer of daratumumab for this purpose. I mean, I guess this was one of the purposes they did. So that was really good. And so magrolimab was another antibody that I talked about, and it interferes with a pre-transfusion testing in a similar way. So this paper here highlights how it does, and I'll just summarize what this paper basically described. And it's CD47 is highly expressed on red blood cell. So 3+ to 4+ is the grading scale for used to measure agglutination, and 4+ is actually the highest of the scale. So that comes to show how strong the reactions are. And CD47 forms a part of the RH macrocomplex on red blood cell, here shown on the top right picture. And this is CD47, and these are the RH complexes, RHD, RCE, and RHAG. And why these complexes are important, because they carry they carry the antigens of the RH blood group system, which has the 56 blood group antigens. And so RH is also it's typed alongside ABO, so it's very important. And so that's where CD47 is, very close to them. So we perform, so it reacts, so being close to the RH molecules, this paper found that it reacts more strongly with red blood cells that were D positive compared to when it was D negative. I thought that was interesting. So this graph here, this table here on the right shows the red blood cell phenotypes that reactivity of patient plasma samples undergoing magrolimab therapy for 4 patients. And I'm sorry, actually, I didn't think about these red blood cell phenotypes. They're written in the blood group nomenclature. So, but basically, yeah, what you can see, so magrolimab reacts with everything because it's on all the red blood cells. You don't see it here because this is just an immediate spin. So you add the plasma to the antibody and then you spin it straight away. You don't wait for it to incubate. So you also don't have a secondary antibody, but the fact that you have very strong reactions without an incubation and without a secondary antibody comes to show that this is that's very high affinity and strongly reacting antibody. And so when you add the 37-degree incubation, so that's about 15 minutes, and then you add a secondary antibody, you get 4+ reactions, strong reactions with the other as expected, but you get strong reactions where there was no reaction in the immediate spin. So it's just a more sensitive assay, and it's actually just detecting it more. But so this ortho anti-IgG reacts with its secondary antibody that reacts with all IgG classes. With this gamma clone anti-IgG, it doesn't react with the IgG4 subclass, and you can see that it's actually removed that strong reactivity that we showed. It removed it almost completely. So that's comes to show how much interference this magrolimab antibody had on the pre-transfusion testing. So what these authors recommend is that we perform the pre-transfusion testing before the start of their therapy. And so we can then record any atypical antibodies present. And so we have those results that we can use to find them compatible blood if they require transfusion during their magrolimab therapy. And so the strong reactivity with red blood cells is not just consequential to the blood bank laboratory, but it also reacts with the patient's own cells. And so after magrolimab has been administered, patients had initial drop in hemoglobin, and that was actually expected. And it actually resolved after about 21 days. So it's something that can build back up, which is kind of good. So magrolimab also has an effect on platelet testing because it's also highly expressed on platelets, and it's not something that I've seen with DARA. So this is something that was quite new for me to learn. So similarly with the red cells, patients had thrombocytopenia, infrequently occurring. So they've also had a drop in platelets after they've been administered magrolimab. And so the routine assay that's used for platelets is called the Capture-P assay, and that detects the platelet-specific antibodies and HLA antibodies in the patient's plasma. And the other one is called the PAC+, which is the solid phase ELISA with wells coated with the antigens of interest. So these are just platelet glycoproteins or HLA class 1 glycoproteins. So this table shows the 8 patients that they tested for this study. And I've shown the test procedure under the results actually. So for this Capture-P assay, the way it works is the platelets from 13 different group O donors were coded onto the well. And then you add the plasma serum. The list is the buffer, I suppose. And so then, yeah, you add the patient serum and then you incubate and you let any antibodies react. So you wash it and then you add the indicator cells. And so if there were, so the antibodies from the patients would just block the sites of any binding. So if it was positive, you wouldn't really get, you won't really get a strong color, but then if it was negative, then you get a strong color in the center because they would just, there wasn't like anything to stick on. And so then you also have a weak, so it's kind of analogous to a competitive ELISA if you've ever done that. And then, so the results for that is like, so they've also, for magrolimab, 100% positive. So that's, but then they've also compared it to DARA. Sorry, oh yeah, take that back what I said. Yeah, so they've also tested for DARA and it's also 100% positive. So DARA and magrolimab interferes with this Capture-P assay. And so I guess you get variable results with the Capture-P ready screen, but with the PAC+, it's actually more specific and you don't get as much of you actually get a more truer result. And where there's a negative, like where there was a positive with the Capture-P assay, you can get a negative with the PAC+, maybe because it's more specific. Happy to hear your thoughts about this because if you also have any comments, because like I think that's very interesting that there's a discrepancy, and it's probably because the PAC+ has a more specific range, like they have fewer targets on the wells, and so you can have more specific antibody binding, whereas the Capture-P assay, it just has the platelets and HLA antigens, and it's a pool from 13 different donors. So that's probably where this these false positives are arising. So I thought that was really interesting, and yeah, So in conclude, like just to wrap up, there are with this section, there are mitigation strategies, and I think with something that we'd only use when an urgent transfusion is required, and if the blood bank laboratory isn't aware. So it's really good for the blood bank laboratory to be aware, so that they can prepare, and also it would provide better outcomes for the patient. And also for smaller blood bank laboratories without access to a reference laboratory, like if they really needed to find blood, they could try these different mitigation strategies for anti-CD38 and for anti-CD47. So yeah, but even then, it's like not the end all. There's other things that we can do. We could genotype the patient, and also, so it's not like, yes, it's a challenge, but it's something that we've found our way around it. There's other technologies that we can use to find compatible blood for patients undergoing these therapies. I won't go through this all, it's very specific, but so in conclusion, yeah, transfusion laboratories need to be aware of clinical trials involving monoclonal antibodies which react with red blood cells. And we need to perform pre-transfusion testing on patients before they receive the monoclonal antibody therapy. But we have mitigation strategies and they're available for us to use in urgent cases. So yes, that wraps up the part 1. Let me know how I'm going for time. The second part is, yep, good.
Yeah, still have time, yeah.
Okay, awesome. The second part is monoclonal antibodies for blood grouping. So a big part of what we do is actually like figuring out someone's blood type, I suppose. Yeah, so it's part of our pre-transfusion testing. So on the left here, so part of what we do, and then the most commonly typed blood groups are ABO and RH. And so you probably, if you've given blood, this is also the blood type, blood groups that you know. And so the way we type for it, we can use these reagents in the lab. And if you probably these might be similar to you, like the very colors of these reagents is there like to help us type. And so like if you're typing in the lab, like you would know blue is anti-A, yellow is anti-B. And so they're IgM or IgG antibodies derived from an in vitro culture. It's either mouse hybridoma or human mouse heterohybridoma. And if we do it manually, we add a couple of drops of this reagent into the tube with the patient's red blood cells, and we visualize the agglutination or no agglutination like this. But if we wanted to do it on a mass scale where we're testing thousands and hundreds of donors, we can do it in an automated way. This is an example of the Beckman-Coulter PK7300, which used to be used in Australia, but then they changed, I think. They upgraded, or I'm not sure, but yes. So like, this is the one that they used when I was working in Australia. And what it does is that still the same reagents like you put in, like you've got the anti-A, anti-B, but it's in a microplate, like a 96 well microplate titer format. So if you have a negative reaction, the red cells would center in the middle because it didn't bind to anything. But if you have a positive reaction, it would you get a nice like homogeneous like spread of the red cells. And so what the machine measures is how much light is emitted in the well, and that classifies it between a positive or negative. And anything discrepant between the two will get tested further by the manual method using this method. So yeah. Okay, so yeah, this is what we do for ABO and RH. And in some cases, we even do it for KEL. I think in Australia they do. So KEL is another blood group antigen that's clinically significant, as mentioned, I think with the before. Okay, so when we want to know other blood group, the phenotype of the patient for other blood groups, they get sent to the blood banking laboratory, and we often do it manually because it is not such a common request. So it's only really, so it's used for extended phenotyping, and these blood group reagents are common for common antigens. They're available and we can buy them, and they're not difficult to source. But for rare blood groups, I think for rare blood groups, like maybe like 1% of the population have them, or like some blood groups that are more common in Asian populations than Caucasian populations, like groups like that, we often don't have those blood group reagents, and we often have to use patient anti-sera to type, which is the finite and limited in amount, and they're often stored for years, and they also might not thaw well either. So in those cases where we don't have any reagents, we perform a blood group genotype on the patient, but I think that's limited because genotyping is great. We have the allele, but it's a phenotype prediction. And it's something that we only have confidence in by testing a number of samples over a number of years. And so for some mutations, we have good confidence that they associate with a particular phenotype, but for some mutations, especially if they're new, we don't know. So that's a whole nother realm. But like what's best is we have the antibody, the specific antibody to type for the blood group that's rare, and we also have the allele. And so now I'll go into developing the monoclonal antibodies, and that's actually not as easy as it could be. I guess we have to think about the way antibodies are formed. Two blood groups. Their antigens on the red blood cell surface, and how they come about is from an alloimmunization event from pregnancy or transfusion. So they're antibodies that are made, that are raised against the native structure on the red blood cell. So here is an example of pregnancy is a way that a mother can be immunized to the baby's own red blood cells. So the mother who is negative for the antigen is exposed to the baby's blood who has the antigen, and she makes antibodies. And if they're IgG, they can cross the placenta and react to the baby's red blood cells and make the baby sick. And so that's what we try to manage when we, so that's what we're trying to manage and trying to avoid. And so just think about the red blood cell surface. There are like several different blood group structures. Here are some of the structures that are currently known as blood group systems. And we have ABO here, which are carbohydrates, RH, a multi-pass transmembrane protein, and CAL, which is it's a blue dot here, but it's a single pass transmembrane protein. And it's also linked to other structures on the red blood cell. So they have their own kinks and their own glycosylation structures and like their own post-translational modifications. So that's something we had to think about when we're developing these monoclonal antibodies. So previous studies have looked at monoclonal antibodies using phage display libraries, and the benefit of that is that you can make them from non-immunized individuals, so naive libraries. So I've decided to focus on two particular studies. This one is from the UK, and it's one of the older papers from 1992. So they had naive libraries, and this group screened it for any antibodies like that they got to ABO, to RH, and to KPA. KPB, which is an antigen in the KEL blood group system. Okay, this blood group, so what they found is that they had a lot of binds. They did 3 rounds of panning. They've screened quite a few, and then they found a few, like they found several positive binders. But what I think came out of it was that they found positive binders, but they were also non-specific, like that's a risk. And they only found a few. So out of all those that they screened, they found like this anti-D and a pan-agglutinin, which is like, so it reacts with all the red blood cells, which isn't quite useful. So I guess it shows that there is a high risk of isolating binders which react with other structures in the red blood cell. And I think they use a subtractive panning strategy as well. So even then, it's not easy. And so they've also used papanized red cells. So they've also like eliminated structures on the red blood cell to better expose the RH protein. And yeah, they were successful, but yes, it's something, it's very complicated to navigate around the red blood cell surface environment. And I think this is something that depends on the blood group system because, yeah, it depends because you've got, these are very immunogenic structures. And I think that's why they were able to isolate such antibodies. And these aren't like rare antibodies, like they're quite common antibodies for people to make. Okay, and then on the right is a way of developing an antibody against the antigen Diego-A, which is more common in Asian populations than in Caucasian. And they've also found a high rate of positive binders with like out of 300 clones, they found 188 were positive, but I think they only reported, I don't know what happened, but they only reported 3 positive binders, and that they've focused on SCFV phage number 4. And it's amazing that they even classified this as a positive binder because a 1+ reaction is a very weak reaction compared to the PBS and control. So especially if they've done that on the 0 to 4+ scale. But then again, this is a SCFV phage, so they are known to not react as strongly. So, and this is very similar to like what I got in my PhD. So I think if they reformatted it to a full IgG antibody, they would be getting very strong 4+ reactions. But that's my prediction. So actually a way to like, I think the way to getting a high affinity, high specificity antibody is by getting B cells from immunized individuals. And that's what I did during my PhD. So reported in this paper for making anti-D antibodies. Out of 84 phage SCFV clones isolated, I found 14 positive binders, and 7 of them were unique. And so reformatted those phage SCFV clones to a human IgG1 backbone. And this is an example of a flow graph. And I think in the serology test, it translated to a 4+, but I got very distinct positive, which is the blue and the red, and negative, which is the black, discrimination by flow cytometry. So that's phage display. And then another popular way of developing monoclonal antibodies for this field is by EBV transformation. So infecting the B cell producing the antibody of interest with Epstein-Barr virus. And they've done this. It's a very popular technique used in Japan. And this is an example using monoclonal anti-JK3. And JK3 is an antigen that's common in the Melanesian population, but very rare in every single other population, actually. So it's a loss of the JKA and JKB antigens in the KID blood group system. So that's the urea transporter on red blood cell. So it's very interesting. And then another example of a technique is the single B cell isolation. So what they do, I'm sure if you've heard, yeah, so it's not an uncommon technique, and what you do is you isolate the single B cell that produces your antibody of interest. And this is what the paper did for a blood group and antigen named VEL. And so after they've isolated that single B cell, they were able to amplify the variable regions and get the sequence and then clone it into the cell line that they wanted to. And then they put it on a human IgM backbone. And so they can just use, sorry, my battery is running low. Sorry. Yeah. Okay, back to where we were. So yeah, they've been able to do that for VEL, which is, I guess, one of the new ones, more common in Caucasian populations than other populations in the world. And okay, and so putting an IgM backbone means they can just add the antibody and the donor's red cell, and then they can visualize the agglutination. So what's interesting with these two antibodies is that what we want to use them for is donor screening. So if an antibody is more common in one population than other populations, you have a disparity of what is available in what is available commercially to buy. And so you have different groups around the world making antibodies, which are important for their blood organization to find blood for. So so this group is actually from the Netherlands. And so it comes to show that, yeah, there's more of a prevalent Caucasian population here. So you want those antibodies so you can find compatible blood that is VEL negative. And it's easy. An antibody is quick and easy compared to a PCR, which is the other way that we can do it. But it's, and so here JK3, I guess you could do it using anti-JKA, anti-JKB antibodies instead. But if you have an anti-JK3 antibody, you can put that, you can make that in bulk and test it against the red blood cells, several hundreds of donors' red blood cells, and you could probably get results quite quickly compared to genotyping for JK3, which would take weeks compared to days.
That sounds exactly like what you should be doing. I mean, if they want you to stay for observation, then stay for observation.
So in conclusion, for routine typing, reagents are readily available, and for the common blood group antigens and for the major clinically significant ones that are relevant to all populations in the world. But if we're developing monoclonal antibodies, we need to recognize blood groups in their native conformation on a blood cell, which is not often to do for most technology with like phage display. And so we need to develop the specific and high affinity monoclonal antibodies. We have to derive them from B cells from immunized individuals. And that's approach that is applicable to multiple antibody development platforms. Yeah, and that concludes my talk. Thank you.
Okay, Nisa, thank you for the interesting presentation.
Thanks.
Sangat menarik. Bapak Ibu sekalian, apakah ada pertanyaan? Boleh di kolom chat atau boleh raise hand. Masih ada waktu sekitar 5 menit.
Okay.
Atau mungkin dari saya dulu. I'm just wondering if the apa ya namanya in clinical practice is already, I mean, is it common to do the the testing before the transfusion? So is it quite important, or what do you think?
I think it is quite important to do it to avoid, because oftentimes there is a risk that the blood bank laboratory might not know the patient is on, like it's not in the clinical notes, the patient is on therapy. So I think there needs to be communication between the doctor and the blood banking laboratory. And so just bringing about this awareness. And then, yeah. And I think probably these patients, probably not their first transfusion they've had either. So probably looking at their clinical history is important as well.
Yeah, because like a bit scared. I mean, I'm a bit scared, like if there's not pre-testing before, and then just like the person get the transfusion, and like your presentation is quite complicated, not as simple. I mean,
Yeah.
I was
Yeah.
amazed, and then
Yeah, agree.
and we only know like about, I mean, standard BO and Resus, and then you said like already 44.
Oh, sorry.
44,
Yeah,
right?
that's 44. Yes.
Yes. So, I mean, based on your experience in maybe in Australia or maybe in Europe, is it common they test all of the blood types before they got the transfusion? Or there's
No,
a pre-screening?
they just do the screening, and they just test for the common antigens like ABO, RH, CAL. Unless so the antibody screen is where they test for antibodies to the rarer antigens. So like,
okay,
so like those red cells have their own unique phenotypes. Like they get specific donors with like their phenotype they want. And so if anything comes up on the antibody screen, then they go, okay, we need to look at the other different blood group antigens. And I guess 44 is a scary number. Sorry, you have another question? I'm just going to keep talking.
Yeah. And I'm satu pertanyaan lagi mungkin tentang yang antibody itu currently it's just, I think if I'm not mistaken, still from the donor, right? I mean, not from recombinant, right? So you try to make the map to, I mean, you try to make the map and then you screen from the H display, because currently the antibody just got from the donor, right? For the anti-D.
Ya, anti-D dari donor. Tapi kalau untuk blood group antigen lain, itu seringnya dari orang yang bikin antibody di klinik. Jadi waktu dia hamil, bikin antibodinya, atau terus dia habis selesai hamilnya, dia jadi donor gitu. Karena iya begitu. Jadi untuk anti-D banyak donor, tapi untuk antigen lain itu gak begitu sering. Itu mesti like transfusion labnya mesti approach orangnya, bilang kamu ada antibody ini. Jadi boleh nggak kita bikin monoklonal dari antibody kamu, like ambil B cell dari kamu, terus culture di lab gitu. Jadi mesti ethics-nya juga mesti you need to satisfy the ethics as well.
Jadi so far belum ada yang dari produk dari develop dari ini maksudnya bukan dari donor gitu, jadi buat sendiri. Like you did, I mean, you tried to make the map, right? Bagaimana itu progresnya? Maksudnya apakah untuk kedepannya mungkin kalau bisa dibanding kita ambil dari donor, kalau kita sudah bisa membuat sendiri gitu ya, maybe it will be better and easier to produce, I mean, for the production, for the mass production. So kita nggak usah lagi ngambil dari orang itu loh, nyari donor gitu.
Nggak usah lagi. We can just find sequences
dan bisa lebih murah kan nanti ya katanya.
Yeah,
Mahal katanya itu ada
mahal.
pasien yang pakai itu berapa 1 juta berapa kalau di sini harus, dan
Mm.
itu susah sekali dapatnya. Jadi kalau sudah ada produknya, maksudnya semoga produk kamu bisa dikembangkan dan bisa masuk
Iya,
clinical trial.
nggak tahu itu semuanya tergantung Red Cross-nya. Jadi saya juga bingung gimana. Saya udah I already have the NTD, tapi kayaknya dia orang investasinya sama grup lain sekarang. Jadi walaupun ada NTD bagus ini, dia orang nggak mau pakai. Dia jadi pakai NTD yang dari single B cell isolation.
Yeah.
Saya juga nggak begitu ngerti karena NTD-nya menurut saya sama aja karena itu spesifik, tapi tergantung backbone-nya. Jadi untuk NTD apa yang kurang kita mengerti itu gimana NTD like work in our body. Jadi itu untuk diagnostik maybe NTD udah established, but for the therapy it's very difficult, and that's what I would have liked to pursue after my PhD, but I think Red Cross-nya, like they took a different direction with a different group, and so yeah, begitu.
Iya. Oke. Ini
Iya.
mungkin satu pertanyaan lagi ya, satu pertanyaan terakhir sebelum ke presentasi yang
Yeah.
kedua. Ada satu pertanyaan di kolom chat. Apa dari UNAS ya, dari Bu Tengku, apakah ada kaitan antara overekspresi dan timbulnya kanker? Overekspresi overexpression apa ya pada gen pada antibody.
Kaitan antara overekspresi timbulnya kanker, overekspresi pada gen pada antibody atau pada apa? Good question. So I encourage you, I think the paper said it, but I think it's just associated with the tumor and what the tumor expresses. I'm not sure particularly, but it is associated with prognosis as well. So I think that's more of a cancer biology question, but I think it's very good. And actually, I didn't think about that question, but I would encourage you to read the paper. I can put the link to the paper in the description, actually.
Mungkin bisa nanti di linknya masuk di kolom chat juga.
Yep.
Atau mungkin ya.
Yeah. Thank you.
Yeah. Thanks Manisa presentasinya. Semoga progres anti-D-nya terus sukses. Oke, ya. Ini ada just hoping from our college. Semoga project anti-D-nya berhasil. Oke, Nisa.
Okay, yeah, I'll just answer the question and then I will leave. Okay, I will answer in the chat though, but thank you so much for your time. Thank you, everyone.
Ya, makasih Nisa. Ya.
Makasih banget ya, bye.
Ya.